Hello, everyone, and thank you for coming to my talk. The conventional paradigm of sexual dichromatism dictates that sexual selection pushes males to be vibrant and colorful to attract mates, while natural selection pushes females to be cryptic. However, this binary description of color was formulated on the idea that female coloration wasn't important and ignores the broad mosaic of variables which influence coloration patterns. Coloration arises from a complex concert of selection pressures and a litany of causal links between different life history strategies, mating systems, habitat structures, and climatic variables influence how color evolves and perpetuates. One of the most important factors driving this pattern is light environment. Several studies using categorical metrics of open and closed have found that the amount of light in a habitat impacts signaling modality and how species can perceive one another. However, using these broad variables are great for displaying broad patterns, but habitats are heterogeneous and complex landscapes, and these binary traits might not capture the true complexity of these habitats. So I'm really curious, how have natural history and traditional habitat metrics influenced patterns of sexual dichromatism? And two, how does habitat heterogeneity influence coloration patterns? So to tackle this question, I use the cardinals as a focal group. This is a fantastic family with a wide range of plumage variability, as well as differing degrees of sexual dichromatism. Additionally, approximately a fifth of the cardinals are migratory, most are sexually monogamous, and they occupy nearly all habitats across the Americas, making them an excellent group to study how habitat structure influences coloration patterns. I traveled to museums across the country where I collected spectrophotometry data for all recognized cardinals from 5 males and 5 females, and for each individual, I measured 17 different standard regions. So I processed this data within tetrahedral color space. I then ran a phylogenetic and a non-phylogenetic PCA. PC1, for the non-phylogenetic PCA, PC1 represents more complex plumage, and PC2, or the negative values of PC1 represent less complex plumage. For the purposes of this talk, I will be using the non-phylogenetic PCA so that male and female plumage are on the same axis of comparison. I then generated a time-calibrated UCE phylogeny for all my phylogenetically controlled analyses. This is that phylogeny. If you want to know more, please come to my poster session on Wednesday. I then incorporated ecological traits of strata and migration, average mass, and 3 different categorical metrics from different data sets. And briefly, I actually found that these data sets didn't match up. And so it really implored on me that I really needed to include a more robust framework to understand how habitat influences coloration patterns. So to do this, first I generated ensemble species distribution models. And these are just distribution models that are a weighted average between 2 different algorithms. Then I took these new distribution models and uploaded them to the Google Earth Engine, which allowed me to align them to a lot of different geospatial data sets and remote sensing databases. One of the primary ones I used is NDVI, or the Normalized Difference Vegetation Index. This is a metric used to understand how vegetation composition changes across both space and time. And it's calculated by comparing 2 different spectral bands, the near infrared band and the visible red band. Briefly, greener habitats reflect a higher proportion of near infrared light. They have more chlorophyll content, and browner habitats reflect a higher proportion of visible red light. They have less chlorophyll content. So you take the difference between these two and you can generate NDVI. And this is a visualization of what that looks like over time. On the left, you'll see a desert dwelling species where you can visualize the ebbs and flows of the seasons, where on the right side of the screen, you can see a tropical forest species where NDVI is much more stable throughout the years. So to analyze these vegetation indices in a phylogenetically controlled manner, I query data on NDVI, its sister band EVI, which has enhanced reflection over the tropics, and then the bands used to calculate them, visible red and near infrared. And then for all 4 of those metrics, I used each of these species range and I queried all 4 of these metrics and generated image stacks based on 16-day intervals from 2000 to 2019. I then took this sandwich of data and reduced it down to a single plane, which allowed me to calculate summary statistics such as mean, standard deviation, min, and max. However, although these are really powerful tools to understand how vegetation composition changes over the years, they can struggle to differentiate between really green forests and really green grasslands. That's because both those habitats can reflect a high proportion of chlorophyll content during certain times of the year. So to get around this limitation, I included 2 additional data sets. The first was percent forest cover. Here I summed all trees above 5 meters and then standardized by their surface area to create a percentage. And then the other data set, I used 17 different land cover types, which after standardizing by their range, I created a percentage per land cover type per species, which I then ran through a PCA. And then finally, to understand how climate influences coloration patterns, I used WorldClimate's bioclimatic database and aligned their 17 climate variables to each species hypervolume centroid, and then I ran a PCA on that. I'm going to go over the results briefly of these 3 data sets. For the land, this is the scatter plot of the land cover PCA. It is colored by percent tree cover. PC1, unfortunately, was describing a lot of noise and variation in the data set. However, PC2 was really informative because it is describing a gradient from explicitly evergreen broadleaf forests to all other habitat types. So now I have a really great trait describing habitat specialists to habitat generalists. This is the climate PCA scatter plot. It is now instead colored by latitude, where greener values are more northern climates and redder values are more southern climates. PC1 is describing a gradient from more seasonal climates to more stable and rainy climates. And climate PC2 is just describing a gradient in precipitation. So I know that was so much, but since most of these traits were likely autocorrelated with one another, I removed all traits that had a regression coefficient of plus or minus 0.75. This resulted in 14 remaining traits. And I implemented a PGLS framework to understand how multiple aspects of plumage are correlated with these different ecogeographic variables. And to do this, I analyzed all single, double, and multi-trait comparisons. And then I ran a PGLS NOVA to understand the relative importance of those variables. So for the results, first I'm going to talk about brilliance, and then I'm going to talk about PC1. So for brilliance, I really just found that birds near the tropics are darker and birds in more temperate regions are lighter. So darker birds have evolved in areas with more precipitation, more forest cover, and if they live in low and light environments like the understory, well, lighter birds have evolved in more seasonal habitats and climates and in areas with less forest cover. PC1, color complexity, had a lot more interesting information though. So for males, I only found a correlation with climate, where males that live in more seasonal habitats have more colorful and complex plumage, and males that live in more stable and rainier climates have less complex plumage. I found no direct association between elevated levels of plumage complexity and habitat heterogeneity. And this actually implies that sex-specific selective pressures are likely driving coloration on a per species basis. Females, on the other hand, had a lot more nuance to them. So across all my comparisons, I found interesting patterns such as females that are larger are more colorful, but across all my regressions and comparisons, one trait consistently had the strongest AIC, and that was standard deviation in visible red. And the very critical component of this is that this trait is not just describing a spatial pattern, but really critically a temporal pattern. Many habitats exhibit a cyclical flow of vegetative greenness, which varies over the seasons. This flow resembles a sine curve where half the year it's greener and the other half of the year it's browner. So these seasonal habitats have these cyclical flows to them. And because even though these habitats can vary dramatically in vegetation structure over the course of a year, when you expand that out to multiple decades, we actually find that habitats that have these routine changes have less variation over time. And that means that female cardinals have likely evolved to be less colorful in these more predictable habitats. Alternatively, female cardinals that are more colorful have evolved in habitats with more variability. These patterns were vis-red changes across both space and time are correlated with more complex plumage. And this shows that elaborate female plumage has to simultaneously balance the compounding pressures of both sexual selection and habitat constraints. And this means that female coloration is far more complex than we previously recognized, and a lot more research is warranted to understand how social selection interacts with female coloration. So in summation, I found no direct association between plumage complexity and habitat structure, other than the general motif of darker habitats produce darker birds. And this implies that sexual selection is likely driving coloration on a per species basis. Female cardinals, on the other hand, were heavily influenced by region-specific ecogeographic effects, where more complex females occur in areas with more variation in visible red. If they are larger and if they are sedentary, well, less complex plumage has evolved in habitats that are seasonal or darker, in birds that are smaller, and in females that are migratory. And this shows that species habitats are not monoliths, that habitat heterogeneity exists on a spectrum rather than a binary ultimatum, and future research needs to incorporate these geospatial data sets in order to understand the true evolution of plumage coloration. And so with that, I'd like to thank my advisor, Kevin Burns, and all the past members of my lab. I'd like to thank the museums that let me collect this color data, the resources that funded this research, Birds of the World for allowing me to use those illustrations that you saw, as well as Daniel J. Field for providing the images that you saw. Thank you so much. And with that, I will take any questions that you have.